population genetics and gene flow in a tundra sedge with tussock cottongrass in the context of climate change and local adaptation. So, some climate change statistics. The mean velocity of climate change is projected to occur at 290 meters per year in the Arctic by the end of the 21st century. And over the last 40 to 50 years, a local temperature increase of 0.5 degrees Celsius per decade has occurred. And this increase in temperature impacts species ranges, phenotypic plasticity at the population and individual level, and then of course evolutionary adaptation to these modified conditions, in this case temperature increase. So here is tussock cottongrass. It's a dominant plant of the moist tundra in northern Alaska. Its mound-like growth habit increases nutrient cycling and also creates microclimates which benefit other organisms such as mosses and lichen. And a 30-year reciprocal transplant experiment was set up along this latitudinal gradient in north central Alaska. 6 populations were chosen. They're split in half by the Brooks Range, so creating a northern component and southern component. Common gardens were set up at each of these populations, and what was found was home site advantage for flowering, fruiting, and survival. And then also, for southern populations transplanted northward, there were higher survival rates than northern populations transplanted southward. So with evidence of local adaptation, also slow growth rates, very low seedling recruitment, what is the potential of this organism to survive as the climate warms? That's what we want to really consider. So a paper by Bennington et al. Kind of lays it out a bit for you. So here you have the mean number of flowers per tussock. In some southern populations, it was more than a 10-fold increase of mean flowers per tussock in the home tussocks versus away tussocks. And then also in your southern common gardens, the northern plants really dropped off in survival rates, especially after 15 years of this reciprocal transplant experiment. So there's also trends in growth rate, northern southern components, flowering, the southern populations senesce later, and then of course survival dropping off for northern plants in the south. So for gene flow, as the tundra warms, northern plants tend to not be able to survive well, and this is especially a problem as shrubs increase in abundance. They're likely not able to compete also with this extra stress there. And we really want to know what the potential is for southern genes to move northward. What are the levels of gene flow occurring in what direction? And also, what is the potential for range expansion? So investigating transcriptome expression levels as well will reveal how plants are responding to environmental cues and just how they're dealing with these changes in climate. All right, so we're doing using NGS sequencing, DDRAD, and are trying to look at the genetic structure along this longitudinal gradient. Will ecotypes be recognized that have been identified in previous and current research, or is it really just a northern and southern component on a population structure level? And then also, of course, is it geographic barriers limiting gene flow? Are they ecological barriers or isolation by distance? And then, of course, are differentially expressed genes providing evidence of adaptation. So, Brett just laid it all all out for you, methods. 14 populations were selected along a longitudinal gradient. VD-rad and stacks provided reed assembly, and we dropped down after some stringent filtering from 50,000 to a little over 5,000 SNPs, and then used admixture for maximum likelihood analyses, and then also additionate and a discriminant analysis of principal components to further look at the genetic structure, and are looking for basic diversity statistics. Here's a northernmost population, just to give you a visual. The tussocks are pretty much, they look dead, and they're just dwarfed, versus coldfoot, a healthy southern population where the spikelets can pretty much reach over 3 feet. Little tussock there. So, some results with admixture, and it was confirmed with our DAPC. We found 3 distinct clusters ranging across this latitudinal gradient. First, Eagle Creek, just, unique cluster, no doubt about it. And it's also a high elevation population at 770 meters, much higher than the other populations along this gradient. And then a southern cluster occurring here, and a northern cluster. What was particularly interesting is previous research suggested the Brooks Range as a barrier to gene flow, but the way these populations clustered is actually suggesting tree line as an ecological barrier to gene flow. A couple populations on the south side of the Brooks Range, outside of the Brooks Range, clustered with the northern populations. So here, I analyzed outliers with Locitan, and then created a neutral loci data set and outlier loci data set, ran the same analyses, and found the same affirmation of 3 clusters across this gradient. So unfortunately, with our population genetic diversity measures, they were largely similar across the populations, and we're currently waiting on some data that's really going to help, because there'll be 10 additional intermediate populations, have more individuals per population. It can be a real pain to find loci that are represented in all individuals across all populations with DB-RAD sequencing. I don't know if anyone else here has experienced issues with that, but the day is coming, and of course with previous ecotype studies, the South has been suggested as being more plastic, and as glaciers receded in the north, there would have been colonization towards the north. There should be less diversity northward. And a preliminary assessment of gene flow also revealed no strong patterns, so that's inconclusive at this point. What's more exciting is the evidence of adaptation at a genetic level. So did some some RNA sequencing. And 6 populations, as indicated by these blue stars, were chosen, and they were transplanted into a common garden at Tuluk, just north of the Brooks Range, in 2012 and 2013. These plants were sampled in last July 2016 on an 80-degree Fahrenheit day, which was a record-setting day at that site, and then also at a 55-degree day. They're only 3 days apart from each other. And then we did RNA extractions. 3 individuals were pooled for each population, and used Trinity to assemble reads into contigs and also look for differentially expressed genes. So what you might expect is a lot of genes, especially that were related to heat shock proteins, were upregulated on the 80-degree day across all populations. And then also a totally different, broad array of completely disparate genes were upregulated in the southern populations in the common garden versus the northern populations in the common garden. So here, I break it down more in the next slide, but you can see that the northern populations were upregulating genes in response to stress, defense, and also heat shock. And then in the southern populations, more genes with cellular components and molecular function associations were upregulated. I broke it down a bit more. So here in the south, you can see that actually water stress genes were being upregulated at more than threefold times more than the south versus the north, and also different genes in relation to stress, such as isoflavone reductase, were being upregulated in the south, and then in the north, we have a broad array of heat shock genes being upregulated, also ethylene, which can also relate to stress response, was upregulated. So basically, the northern population seemed to just not be able to handle heat at all and are going into heat shock, while the southern populations have other methods of dealing with extreme heat. It's not totally alien to them, so it's pretty exciting, and we're going to further look into this transcriptomics of adaptation. So at this point, there's genetic structure along the longitudinal gradient in northern Alaska, and it seems that tree line is an ecological barrier. The structure analysis supports a genetic component to these ecotypes, and then we found Eagle Creek as its own unique cluster. And then transcriptomics provide evidence of local adaptation, which is especially exciting for further Arctic applications and looking at other Arctic plants. Future research is get those other individuals and more populations, especially more thorough sampling north and south of the tree line, and get gene flow modeling going, look to depth at gene flow analyses, and then at the transcriptomics of adaptation. So these are my acknowledgements, and I'd be happy to take any questions. Thank you.